I would like to welcome everybody to this Q&A webinar to discuss the CFP for CHIM for beta coronaviruses. And today we are going to address the call text and to address some questions you have, and so that you are more clear in what we expect from the application and what we expect from the program. And yeah, I'm glad that you have the interest in this funding program and in the CHIM program. And before I start with the webinar, I would like to give some housekeeping rules. The housekeeping rules are that this webinar is being recorded, and this webinar will also be shared on the TJHN platform. And if you have any if you want to introduce yourself, or if you have any technical issues, please use the chat box. And if you have questions which will be addressed later on, please use the Q&A box and to post your questions. And Luz, our project manager, will later on read the questions if we have time, and then we will discuss the questions. Due to the number of the participants, your camera and microphone are disabled, and so if you have questions, we will read the questions and discuss them later on. And there may be an opportunity at the end of the Q&A session to speak, but I think Luz will have the only availability to use the to speak, and so that we can be a bit smoothly to discuss all the questions and answers. And please note that all the questions we address during this webinar, we will also write them down and document, and this document will be uploaded and will be made public next week. I hope early next week. So you will have all the answers and all the questions we discussed today next week. And if you have also questions after the webinar, please write an email to us or reach out to us, and we will add these questions in this document. So let's start. And the agenda is like that. I would like to welcome you and also to discuss a bit CEPI's background, why we are going to have call for CHIM. And then I would like to delineate the call for CHIM for the beta coronaviruses, our timelines, and then we have questions already received from some of you. And then in the last section, we have time for open questions, what you can address in the Q&A box, please. So CEPI has a vision and mission and strategic objectives, and I would like to highlight them to bring them into the context of why we are going to have this CHIM call at the moment. So the CEPI is a global partnership, and the vision from CEPI is a world in which epidemics and pandemics are no longer a threat to humanity. And the mission is to accelerate the development of vaccines and other biologic countermeasures against epidemic and pandemic threats, so they can be accessible to all people in need. And CEPI was founded in 2017 and has specific strategic objectives. And I've now listed here 6 of the objectives, with the first one, which is about strengthening defenses against COVID-19 to reduce the risk of future coronavirus pandemics. In the second objective, we want to accelerate the development of vaccines and other biologic countermeasures for known threats. Then the production of library of prototype vaccines, transform vaccine manufacturing. The next one would be to scale up enabling sciences and establish global networks for lab capacities, assays and preclinical models. And then finally, as a 6th objective, to improve coordination of global vaccine R&D and enable equitable to life-saving innovations. So CEPI has made significant fundings and has a growing R&D portfolio, and it was founded 2017, and since then we have about 21 vaccine candidates in our portfolio, 3 rapid response platforms, and also multiple enabling science projects. And starting from 2020, we have now 14 COVID vaccine candidates in our portfolio and also established a centralized lab network and also an animal model network. And now we are also increasing our calls, and there are always new open calls for proposal on our webpage, which you can read and also update and apply for them. We have now a new open call, and this is the reason why you are here at the webinar, because we have the controlled human infection model for Beta coronavirus call. And this will be a new area for CEPI. And CEPI thinks that a CHIM model and CHIM proposal will be beneficial for vaccine development, as it can improve the vaccine development and also focus on mucosal immunity and also mucosal vaccines. So CEPI has announced a call for proposals with the aim to build a consortium to conduct the controlled human infection models for beta coronaviruses and to perform vaccine trials using the established models. And the program will be co-funded by the European Union. And with this, I would like to start the call text and go quickly through our idea why we are doing this call and what is our expectation on this program. So this call is co-funded by the European Union and within the framework of HERA, the Health Emergency Preparedness and Response Authority. And this is the central element for strengthening the European Health Union and the preparedness and response to serious cross-border health threats. And it will enable rapid availability, access, and distribution of needed countermeasures. And CEPI and HERA, the European Commission, have agreed for joint funding projects. And this was signed in 2019 to finance and coordinate the development of new medical countermeasures to prevent and contain epidemics. And with this, the CHIM call is now one of the projects which is co-funded by HERA and CEPI. And we believe that using a controlled human infection model by using a known doses and a known time point of inoculation and a really comprehensive and comprehensive safety and immunogenicity monitoring, we can learn a lot from this from CHIM models for our vaccines. So CEPI has wants to use the CHIM because CHIM offers the possibility to investigate vaccine-induced mucosal immune response and vaccine efficacy against infection. So we want to focus on mucosal immunity, and if vaccines are able to prevent infection, then the CHIM may open new options also for the development and approval for novel vaccines. When we think about the intranasal vaccines and further on, we want to use them to compare also the vaccines or multiple vaccine candidates, and then maybe also select the most promising or deselect some of the vaccine candidates. So we want to focus on a CHIM-based vaccine trials. So when we think about human challenge studies, we think that a well-designed human challenge study provides one of the most efficient and scientifically powerful means for testing vaccines, and that a CHIM can support the identification of correlates, for example, and investigate the risk of transmission, and also to evaluate mucosal immunity comprehensively. So what is the rationale behind the project? So we think that there are current challenges in the current situation and in the context of pandemic preparedness. So currently there's a lack of licensed transmission blocking vaccines or in the EU, and these transmission blocking vaccines are known or should be able to prevent infection and then may have an impact, a huge impact in the virus spread in the human population. Therefore, we think that transmission blocking vaccines might be critical for the preparedness innovations. Then We lack harmonized, validated research models to evaluate the efficacy of these kind of transmission blocking vaccines, and we lack informative, standardized, and qualified assays to monitor mucosal immunity, and we lack immune markers associated with reduced risk or even blocking infection and transmission. So therefore, we think that there are a few research priorities we should focus on. And one of them is the understanding mucosal transmission. We think that there are huge knowledge gaps in the understanding of mucosal immunity and how the virus is transmitted. Then identifying immune markers that are associated with protection against infection and transmission, developing concepts to measure transmission blocking potential, and then finally also to develop vaccines that are able to prevent infection or transmission. And we think that the CHIM might be a good tool to address these research priorities and also then to address the challenges which we have at the moment. So what can we learn from the SARS-CoV-2? And we think that SARS-CoV-2 might be a good model to use for the CHIM to investigate into the mucosal immunogenicity. So understanding mucosal immunogenicity with the aim to identify immune markers. We think that understanding SARS-CoV-2 transmission is critical, and with the aim to have a tool to measure transmission blocking potential of vaccines, and then to understand mucosal vaccines with the aim to prevent infection and transmission. So mucosal vaccines are thought or suggested to block infection and transmission, or we hope that mucosal vaccines can block infection and transmission, and therefore may influence the epidemic curve rather than disease load. And we observe that there are an increasing number of new COVID-19 vaccine candidates that target mucosal immunity, but there's still a lack of consistency to measure the vaccine-induced mucosal immune response and also the potential on prevention of infection. So therefore, we want to look ahead and want to establish a collaborative multi-country network that establishes kind of a platform for testing mucosal immunity and the transmission blocking potential, and therefore might have the impact to accelerate the development of novel vaccine candidates against coronaviruses, other respiratory viruses, and also when we think of future disease X, and here thinking of the respiratory disease X. So, and we have, we expect some outcomes, and we think about 3 pillars that where we should focus on. So on the one side, of course, we have the CHIM model, and here we want to have as an outcome harmonized trials that can be conducted. We want to have a library of virus stocks available and which can be shared globally. We want to investigate the mucosal immune responses, and here develop an analysis plan and also a guidance to measure mucosal immunity and gain knowledge on vaccine-induced mucosal immune response and about how mucosal immune response can stop infection or prevent infection and transmission. And finally, also to focus on the transmission blocking vaccines or mucosal vaccines. And here again, gain knowledge on these kind of vaccine candidates and also hopefully identify some correlates or immune marker or immune signatures that mediate prevention of infection. And these 3 pillars should be included in this toolbox and in the network and connected to each other. And with this, we think that we are better prepared for respiratory disease X and also for potentially novel coronavirus viruses. So for CEPI, this is it's a new area, and we have also some colleagues that are always that are working together on this program. So we have a core CHIM team and the project lead. I'm the project lead, and we have Luz as a project manager, and we have Alex as a project coordinator. And we have the different departments at CEPI, and we get information and also guidance from all the different departments. So we have the clinical development team, where I'm included. Then we have colleagues working in the laboratory research innovations department. They are also involved in the CHIM team. The innovative technology colleagues are working with us, regulatory CMC, but also external networks like the animal network, the centralized labs, and also speak from the Brighton collaboration will all be involved in this program and assist the successful applicants with the program, with the procedures. If there are challenges, we are all working together on this program to have a successful program, and also to review the application. So we have a huge a team also from external partners that will review the applications and guide also the program and give input. And yeah. So coming now to the CFP. So the CHIM program has the main aim to build a collaborative multinational or multi-country network, because we want to develop a coronavirus CHIM, and we want to have a really multinational network that has an impact on all different levels, starting from virus isolation until to the infection model, and then entering the vaccine trials. And during the vaccine trials, we want to focus on innovative approaches, how to measure transmission blocking potential, and also to investigate mucosal immunity, to establish some assays, harmonized assays, and develop criteria, which can then also be used by other research groups and also for other clinical trials. Okay. And we have specific work packages, and here are 6 pillars shown, starting again from the challenge virus stock in the human infection model, the vaccine trial, mucosal immunity, transmission, and data science. And all these 6 pillars are included in our 5 work packages shown on the right side. So we have work package A to E, and all these different work packages focus on the preparation and the conduct of the CHIM, and then also on the vaccine trial. So we have the release of the challenge virus, to develop a CHIM for beta coronaviruses, to conduct the CHIM to test vaccines, then to monitor mucosal-mediated immunity, transmission blocking in humans, and then also to have the statistics in systems biology. And this graphic shows you that we also expect to have 2 work streams during this program. So we will have a 60-month program starting from January 2024. And then we have in work package A, the virus change agent production and the release of the virus change agent, which will then goes to the clinics to develop an infection model. And if we have then a successful infection model, goes to the vaccine trial, and doing also in parallel the mucosal immunity and the data science projects. On the other side, we already might have challenge agents available, and therefore we can directly start the infection models and then go to the vaccine trial. So this will be at 2 work streams, and therefore not to have a delayed process of the vaccine. The deliverables will be to have specific data packages, but also to have criteria and principles and guidance how to conduct the CHIM and the vaccine trials, and then also to share protocols and reagents. So coming to our objectives, which are also written down in our call text, but I will go quickly through all the objectives so that we can discuss it later. So the general objective is, and the major aim is, to build a collaborative network to develop coronavirus CHIM for testing the vaccines. And we think that independent ethics boards are really an important issue, and each contracted consortium needs to have this in place. Further on, we expect the consortium to develop an ethical framework and an ethics review process, and also to collaborate and communicate closely with the national regulatory and ethics committees and authorities, and also with the public health organizations. Furthermore, we also expect the consortium to advance the communication and to have a community engagement, and to share also, again, the reagents and procedures to harmonize studies globally. And in this context, also to disseminate results and concepts through publications, and also develop strategies to sustain the consortia beyond the funding period, because this, I think, is also the main aspect, not only to have these 5 years, but what can we do afterwards? Can we fund, can we expand the program, or can we fund other projects afterwards? So for the first work packages, we want that the consortium develop guidelines for selection, isolation, growing, characterization, production, and release of the human virus changed strains, and then have also innovative approaches to be tested to accelerate the manufacturing and release of virus stocks. This is desirable but not mandatory. So, and then we want to have established harmonized procedures to conduct CHIM, and also then to have the vaccine trials, and to really focus on mucosal immunity and how can we measure the risk of infection and transmission after vaccination. So coming to the next work pictures and the objectives. So we really want to focus on harmonizing the assays and criteria for monitoring mucosal-mediated immunity and also transmission blocking potential induced by the vaccines, and then also to develop guidelines for measuring vaccine-induced mucosal immune responses, and to develop concepts to investigate vaccine-induced transmission blocking potential, and then also to investigate the strategies and develop strategies. How can we find correlates or surrogate markers? Furthermore, the consortia should develop methods for high-dimensional data investigations, and also how to investigate immunological signatures, and how these signatures correlate or associate with protection against infection or transmission, and then develop strategies for using simulation models to assess the impact of vaccination, and finally, to develop concepts for statistical methodology, and to evaluate data and identify immune markers, immune signatures, or correlates of protection against prevention of infection and transmission. So after you submit your proposal and your application, the first step from CEPI's side is to investigate if these applications are eligible, and if we have, if the institutes or if the capacities and facilities are eligible for this program. And this will be the first step in the procedure of this funding program. So eligibility criteria are for the lead partner. So there will be one lead partner, is an in-depth experience at CHIM trials and or vaccine trials, and also to have the appropriate qualified facilities available, and then proven track record of managing complex clinical trial consortia, and also to oversee and also guide decision-making processes that provide regular reports to CEPI and its partners. If there are clinical trial sites, they need to show experience, and also that they conducted already CHIM studies and vaccine trials, and that they have a facility which has a BSL-2 or better BSL-3 facility. Then thinking of the biotech companies or academic institutions or universities, depending on the part they are applying, so they need to show if they have biobank expertise to provide, for example, the clinical virus isolates. They need to show expertise in coronavirus life cycle replication pathogenesis measurement, and also to showing that they have the capacity and the facility to grow, characterize, and produce the virus change strain. And here we, so there need to be a GMP-like facility and also BSL-3 facility. And the next, the biotech companies and academic institution or university hospitals need to provide evidence on the experience in monitoring vaccine-induced mucosal immunity and also assay development and their expertise in and the capacity to measure viral transmission. And finally, for the biostatistical part, they need to show their experiences in analyzing multi-dimensional data. So the WERVI criteria, I don't go through these WERVI criteria because they are it's really comprehensive, and I want to have time for the Q&A later on. But these WERVI criteria are listed in the core text, and therefore you can read them in the text. And if you have questions or concerns, please reach out to us, and we can discuss the criteria in more detail after the call or after the meeting. So these are all the different WERVI criteria which we are going through, and which we will then score the applications and will decide if this application is successful or not. Please be also aware that each award must adhere to CEPI's policies, which can be found on the CEPI's website, and any funding is dependent on the signing of an award agreement, which provides the terms and conditions under which the award will be made. And this is in line with CEPI's third-party code. So if you're going to apply, please have a look to these CEPI's policies, and please take your time and consider if your institute or your company is willing to align with these policies. So coming now to the timelines. So the application submission deadline is the 15th of September at 5 o'clock CET. And afterwards, we have the first step will be eligibility. And then if the applicants are eligible, a peer review will start, and the selection will be then in the end of this year. And we will have a due diligence and contract signature, hopefully end of the year. And a kickoff meeting is planned for January 2024. And then we have a grant duration of 60 months. So coming now to our frequently asked questions. So you provided some questions already, and I listed them. Some questions were often asked, and I summarized the questions. And so I hope I have now a complete list of questions. If not, please reach out to us or write some questions into the Q&A box. So the first question was that there's a total length of the application, about 20 pages. And the question is that if the 20 pages refer only to section 5, which means project summary, or if the 20 pages must be all section of the application templates fit within the 20-page limit. And yes, it should fit into the 20-page limit. The rationale behind this 20 pages is that this first round of application is really a high-level application round. So we want to investigate if your capacities and facilities and institutes and organizations are eligible for this funding, and if the programs has a soundness and the concept is good, and if the timelines and the budget fit into our ideas. And therefore, we think that 20 pages are enough for this high-level concept. And if there's a successful application, we will enter the due diligence process, and then we need more information on the program, specific information on the budget, and then it will go into more detail. But at this first round, we want to have a high-level concept, but nevertheless, you have also the ability to use appendices. So when you think we need to know more, and there's more information needed for your program, please use appendices and then include your concept or your facility descriptions by these appendices. But for now, please keep the 20 pages and try to focus on a high-level concept. But if a consortium responds to all components of the proposal, and including really all parts from part A to part E, and not only one or two parts, does the same page limit apply? So this is a specific case, which I would say, so the application template was indeed designed for specific parts, and we didn't expect that a consortium really responds to all components, and therefore, I recognize that this will be a problem. In this case, these 20 pages will not be enough, and therefore we allow, if a consortium responds in the proposal for all parts, you have a page limit of 20 more pages. So you have the 40 pages, which you can use. But don't overuse it, don't overuse also the appendices. So we need to be able to read all the proposals. So then are the guidelines for the length and the content of appendices and what information may be included. So we want to have it more open. So there are no really rules. So we have the CVs which need to be included in the appendices. If you have information on your facilities, please include these in the appendices, and also your quality standards and so on. This can be all included in the appendices. We want to have, so because we have this kind of 20-page limit, we thought that the appendices can be a bit more open, but please don't overuse it, and keep it focused and readable for us. So the next question is, the call text does not mention the overall budget envelope. And if you want to cover all areas with which overall budget can be planned. And so we decided not to mention the overall budget envelope, because in this first step, we want you to think about what is the realistic budget for your program. And during this first application, we will recognize the budget and we will think about the budget, but after entering the due diligence process, we will then revise the budget and then go into more details. So the first step is really make a high-level concept of your budget. Think about what is the most realistic budget for your program, and then in the next step, we will go into more depth into the budget calculations. There was a question about PhDs and PIs, and yes, you can use the funding for PhDs and also for PIs, but for PIs, we assume that PIs also have different projects, so it's not a full-time PI position which need to be funded, I assume. And also, when you think about 5 years, it need to make sense when the PI works hard or more than on the other years. So please consider your program when the PI is working on this project specifically, and consider that it's not a 100% position for this project. Are there any restrictions on the equipment? So this does not aim to fund capacity. So we don't want to fund really capacity and equipment, but if it makes sense, and if you have a good explanation on the equipment, you can add equipment costs in your funding proposal, and you need to explain why is it needed, what is the benefit of funding this equipment for the consortium, and also when you think about the 5 years funding period. So then of course, when there are plans and they may change because it's a 5 years program, how can we change the budget and how would it take place? So CEPI has mechanisms to change the budget, and nevertheless, although there are mechanisms, it is not the easiest way. So because CEPI governance has to approve it, but it is doable, and we can change budget if it is required. So please make now realistic planning. And if there are changes in the program, we can always go back to the governance and ask for additional funding. So then the question was about what the financial reporting requirements will be. So we have some, so CEPI expect that there's a financial reporting every 6 months, so semi-annually. But we also have every 3 months a meeting called JMAC, and here we need a high-level update on the financial situation. So then the fact is acceptable, so I don't have to add any more information on this, yes. And then if we would like to provide our expertise only for a specific part, and the question was, do we need to have partners or do we name them in the consortium table? So no, you can, if you want to provide your expertise for part A or for part B, please mention it for which part your expertise is needed, and then we will then do the matching with different consortiums or different partners, and we will do the matching procedure, so you don't have to think about it. Then there was a question about the CEPI laboratory network. So does the consortium have access to the qualified immunological tests of the centralized CEPI network? And if the consortium need to calculate this. And so I think it is important to include the laboratory network assays and to use the laboratory network, but not as your own immune monitoring strategy. So these laboratory networks will be an add-on. So to make it more comparable also to other vaccine candidates and to have a more comparable insight into the vaccine-induced immunogenicity. But you will have your own immune monitoring assays and you will have your own strategies, and you just, after you start your program, can apply at the laboratory network, and we will facilitate the analysis, and you can then ask for specific analysis of samples, but this will not be included in the costs and calculations now. But you can mention it in your application that you want to have specific or subset of your samples analyzed by the laboratory network. The same for the animal model network. So here for the CEPI animal network, we have established an animal model network, and they are really experienced and have a high quality and standards of doing these animal testing studies. But if you have your own animal model available, you can apply with this animal model, and nevertheless, the CEPI animal model network will review this your own model and also want to ask you for your strategy and for your study protocols, and then we'll review it and we'll work with you together so that we have a standardized animal model. On the other side, if you have an animal model but you missed some assays, for example, you can use CEPI animal network for specific gaps, so they can fill specific gaps for this approach. And on the other side, if you do not have animal models, you can ask. We can assist that your assays will then be investigated by using the CEPI animal model network. So 3 pathways are doable with a SEPI animal model network. But nevertheless, if you're planning to do animals and models, the SEPI animal model network will be involved in the strategy. Is it possible to increase the number of CVs? So if you, going back to the case when you apply for all parts, then you can increase the number of CVs. And please, nevertheless, please limit the number of CVs because we all need to have to read it, but suggesting like 2 CVs per work package. Are there specific rules for partners and subcontractors for example, for an organization act as both consortium partner and subcontractor? So we do not have a specific rule, and this for me is a new scenario. So you can mention it, so there's no, but this will be really debated and discussed in the next step during the due diligence process. So, and then the legal partners will figure out how we can deal with this situation. Then should we include an Excel budget as an attachment, or can we enter the budget information to the Word application? So I will share you one in the next slide, one slide of how you can use a budget information table. And this, in the first step again, need to be really high level. So we do not need to have really specific information or really much information on the budget. It will be a high level budget table. And the next question is, if we include the budget in Word form, does the 500 word limit apply to the budget itself or only to the narrative justification? And this is only to the narrative justification, so it will not. So if you have it in the Word form, this will not apply for the 500 word limitation. The next question is regarding part A for shared challenge agents. So this will require agreement between regulatory agencies and with a topic about GMP-like or fit for purpose acceptance criteria. And is there expected to be facilitated discussion? So we expect that the consortium reach out to the specific agency and discuss this aspect. We can support discussions, but nevertheless, we would like to highlight that we want to develop a virus library, for example, with a challenge agent library, and this needs to be shared globally, and therefore we believe that a GMP-like manufacturing should be used because other countries have different thinking on the acceptance criteria, and therefore we want to have the highest standard here. Next question about the ethical bodies. Again, this is expected to be initiated by the consortium. And then I have to say, maybe you can also, this person can reach out to me later on. I didn't really get the question. If 2 consortia are successful, what would this look like? So I don't get the question, I think, but I would like to highlight that if we fund 2 consortia, these 2 consortia will not have the same topic. So if we fund 2 consortia, this will be, for example, a consortium for SARS-CoV-2 or for seasonal, or they have specific projects or aims, so they don't really interact or compete against each other. Okay, and this is from Luce, the table, and we will include this in our document, which will be shared early next week. So this is really high level. You can add more information. This is really a high level table, but please keep in mind that we, at this first round, we do not need to have really detailed costs. I think you have to think about it for your budget calculation. You need to think about more details on how to calculate it, but we do not need to know every step. And with this, I finished my questions and want to ask Luz if she can unmute and maybe if there are any questions in the Q&A box.
Sure, Christine. We have 4 questions on the chat, on the question and answer window. So the first one is asking, will the core CHIM team be involved in the research themselves?
No. So it's only guidance, and if you have questions or if you need support in some questions, this CEPI core team will assist, or also if there are some challenges. It's just for facilitating the program and supporting the program.
The next question says, undertaking these CHIM studies on background of persistent circulating and notoriously mutating virus carries some ELS concerns that in case we have a resurgence of COVID from new variants, we should be able to differentiate CHIM viruses from natural variants. It means any design of viruses must include resilient molecular signatures for future recall. The idea is to avoid finger pointing to CHIM study sites as potential sources of leakage.
What was the question? Sorry. Was it a question or was it just a notification?
I think it's more of a comment of the concerns of the CHIM variant becoming leaking into the circulating population, and not.
So it need to have a BSEP-free facility.
Biocontainment.
Yeah.
Okay.
So for the CHIM at the moment, I think BSEP-free facilities are required to do a CHIM and also the vaccine trial using the CHIM.
The next question is asking, how important is the social science research for the CHIM studies?
The social science research, maybe they can add more information on it, and we can answer the question afterwards in the document, which we will share. So
elaborate more. Sure.
Yeah. Please add more information on the questions.
Okay. The next question is eligibility based on historical experience or on demonstrated ability to perform CHIM trials?
So no, it needs to demonstrate the ability. Of course, if they are experienced for several years, it will be a benefit. But the first, for eligibility, it needs to be demonstrate ability. So.
The next question is, we hope to optimize both existing and novel assays as correlates of protection. Will the CEPI network laboratories be able to work with us to develop the best of these as qualified assays, or can we only access already established assays? That's a good question.
Yeah, that's a good question. And I think the centralized lab network will assist and support the development of new assays. So I suggest that, or assume that they don't have the assays available, but they have the experience how to qualify and validate the assays. And so they will work closely together with a consortium.
Okay, we also have some comments on the chat, so I'm going to switch from the Q&A. So the first comment is, what is the readout of vaccine efficacy intended for licensure or proof of concept for later field trials? This can impact the arm size estimates and statistics.
Yeah, so this is a good question, which is not the topic of the application. So we do not need to have this kind of information at the moment, and you don't need to include this information in your application. This will be a topic which we need to work on during the program.
Okay, the next comment is, if only one consortium is to be chosen per coronavirus species, how is this to be decided? Is this based on the chimp facility containment? In a real pandemic, you would need several units to assess vaccines.
Yeah. So the consortium will have to have more than one site. It's a consortium, so we want to include several. It's not only one study site that we will include in the chimp consortium.
The next comment is there a difference on the balance of application strength, i.e. single applicant versus full consortium provision?
No, it depends. So we need to see the applications, and then if there's a single application showing really good and unique opportunity to be beneficial for the whole consortium, we will of course do matching procedures and matching discussions. So there's no problem and bias.
Okay, and the last comment I have is, should CEPI fund 2 consortia, can partners or subcontractors be involved in both consortia?
Yes.
Okay, that's all I seem to have right now, Christine. We have 5 minutes left.
Okay.
Let me see if there is any other. Nope, that's it for now.
Yeah. So then I want to highlight that this document will be shared, hopefully in the beginning of next week, and uploaded at the CEPI's webpage. And we will share it with all of you. And then it will also upload it in the next week. If you have additional questions coming up during the next days, please send us the questions and we will include them in the document. And with this, I would like to thank you very much for your interest in the program, and please reach out to us if you have further questions or concerns, and we are happy to support you in the program and in the application process. And that's it from my side. Thank you very much.